Hi, hello viewers. Welcome back again to one more of the lecture session from this channel. So in this lecture session, we will be studying regarding Meselson and Stahl experiment from the molecular basis of inheritance in detail. So here, prior to the explanation of Meselson and Stahl experiment in detail, at first let us have a look on the mechanism termed as replication. So here, what is replication, or how we can define the term known as replication? Replication, it is the mechanism or it is the process through which copying of genetic material takes place from one generation to the another generation. And most commonly replication, it occurs during the S phase of the cell cycle. So just let me write it here. Replication is the mechanism or process through which copying of genetic material takes place. Genetic material takes place from one generation to another generation. From one generation to another generation. And most commonly the mechanism of replication, it occurs during the S phase of the cell cycle. Replication occurs during the S phase of the cell cycle. During the S phase of the cell cycle, right? And here, after the successful elucidation of the double helical structure of the DNA by James Watson and as well as Francis Crick, within no time, Matthew Meselson and Franklin Stahl in the year 1958, they have carried out one of the groundbreaking observations in order to prove that replication is semi-conservative. Matthew Meselson and Franklin Stahl in the year 1958 have carried out this observation in order to prove that replication is semi-conservative, right? So here, what is the meaning of semi-conservative mode of replication? So here the meaning of the semi-conservative mode of replication is very simple. So during the mechanism of replication, from the parental double-stranded DNA, daughter double-stranded DNA in general it synthesizes here. So just look at here. Let us assume it as a parental double-stranded DNA. Parental double-stranded DNA, right? Parental double-stranded DNA. So from the parental double-stranded DNA, during the mechanism of replication, daughter double-stranded DNA in general it synthesizes here. Daughter double-stranded DNA, right? Daughter double-stranded DNA. Daughter double-stranded DNA, right? So here in case of the parental double-stranded DNA, out of the two single strands, both the single strands from the parental origin, they act as templates here. So it is one template and it is the other template, right? So here both the strands of the DNA from the parental origin, they act as templates in order to synthesize the daughter double-stranded DNA. So here in case of the daughter double-stranded DNA, out of the two single strand, one single strand from the daughter DNA, it remains very similar likewise the nucleotide sequence of one particular template DNA. Is it clear? The nucleotide sequence of one particular dotted DNA strand, it remains very similar. Likewise, the nucleotide sequence of one particular template DNA strand. And the other dotted DNA strand, it successfully synthesizes because of the complementarity relationship. So, such specific relationship or such specific mode of replication, here it is termed as what? Semi-conservative mode of replication. Semi-conservative mode of replication. So here, during the semi-conservative mode of replication, from the parental double-stranded DNA, dotted double-stranded DNA, in general, it synthesizes here. Out of the two single strands, in case of the dotted DNA, one single strand, the nucleotide sequence of one dotted DNA strand, it remains very similar. Likewise, the nucleotide sequence of one particular template DNA. And the other dotted DNA strand, it successfully synthesizes because of the complementarity relationship. So, such specific mode of replication, here it is termed as what? Semi-conservative mode of replication. So, here, in order to prove that the mechanism or the mode of replication is semi-conservative, Meselson and Stahl, they have carried out one of the groundbreaking observations in the year 1958. So, right now we will be studying regarding the experimental proof which was carried out by Meselson and Stahl in the year 1958 in detail. Is it clear? So let me arrange the things here. And if you look at the experimental proof which was carried out by Meselson and Stahl. So initially these people, they have grown up the particular equalizers or the specific equalizers in case of a solution which was enriched with ammonium chloride. So just look at here. So let us assume it as an ammonium chloride solution. Ammonium chloride solution, right? Ammonium chloride containing solution. And here it is nothing but what? Ammonium chloride. Ammonium chloride. And here all these were the equalizers. Equalizers. So here the specific equalizers, by taking the advantage of the nitrogen which is a part of the ammonium chloride, here the equalizers, they were capable to induce the production of DNA. Because as we all know, one specific DNA, here it is nothing but what? Nucleic acid. Nucleic acid, it is composed of many number of nucleotides. And here each nucleotide, it is composed of one specific nitrogenous base. That means, in order to regulate the production of DNA, here what is required here? Nitrogenous source is required. And here the specific nitrogenous source, it is provided by the means of ammonium chloride. And here the specific nitrogen, which is a part of the ammonium chloride, here it is nothing but what? Heavy isotope. In general, the normal nitrogen, here it is termed as what? Lighter isotope, which is nothing but N14. But whereas the nitrogen, which is a part of the ammonium chloride, here it is nothing but what? Heavier isotope, which is nothing but N15. Heavy isotope, right? Heavy isotope, which is nothing but N15. So here, by taking the advantage of the nitrogen, which is a part of the ammonium chloride, here the equalizers, they were capable to induce the production of DNA. So here, after the successful production of the DNA within the equalizers, here these people, they have isolated the DNA from the equalizers. After the successful production of the DNA within the equalizers, by taking the advantage of the nitrogenous source from the ammonium chloride, then after these people, they have isolated the DNA from the equalizers. And here the specific DNA, here it was termed as what? Heavy isotope containing DNA. So just look at here. Heavy isotope containing DNA. Heavy isotope containing DNA, right? Heavy isotope containing DNA. So here, why it was termed as heavy isotope containing DNA here? Because the DNA within the equalizers, it was successfully produced by taking the advantage of the nitrogen, which was a part of the ammonium chloride here. So the reason why the DNA which was obtained within the E. coli cells. Here it was termed as what? Heavy isotope containing DNA. So here after the successful production of heavy isotope containing DNA within the E. coli cells, then after these people they have placed the particular DNA in case of the centrifuge tube which was enriched with cesium chloride solution. Is it clear? After the successful isolation of the heavy isotope containing DNA from the E. coli cells, then after these people they have placed the particular DNA in case of the centrifuge tube which was enriched with cesium chloride solution. And then after the placement of the DNA, after the successful placement of the heavy isotope containing DNA in case of the centrifuge tube which was enriched with cesium chloride solution, then after they have carried out density gradient centrifugation. So in case of the density gradient centrifugation, by taking the advantage of density as the factor, here the DNA band pattern, it gets successfully separated here. And during the density gradient centrifugation, these people they have observed the DNA band pattern which was remained at the bottom of the centrifuge tube. So just look at here. Let us assume it as a centrifuge tube. Centrifuge tube, right? So within the centrifuge tube, these people they have observed the heavy isotope containing DNA which was remained at the bottom. Heavy isotope containing DNA which was remained at the bottom. So here, why this particular DNA band pattern, it was remained at the bottom here? Because the particular DNA, it was enriched with N15. So the reason why, because of the higher density or heavier density, the DNA band pattern, it was remained at the bottom of the centrifuge tube. Then after in the second strategy, again these people, they have grown up the particular E. coli cells in case of the normal nitrogenous source. Initially the particular E. coli cells, they were grown up in case of the ammonium chloride solution. But here in the second strategy, again the similar E. coli cells, they were grown up in case of the normal nitrogenous source which is having the lighter isotope which is nothing but N14. So here by taking the advantage of the normal nitrogenous source, again the particular E. coli cells, they were capable to induce the production of DNA. After the successful production of the DNA within the E. coli cells, then after, again these people, they have isolated the DNA from the E. coli cells. After the successful production of the DNA within the E. coli cells, these people, they have isolated the DNA from the E. coli cells. And here the DNA, here it was termed as what? Hybrid DNA. Hybrid DNA, it is an association of both N14 and as well as N15. So just look at here. And here the generation time, it was around 20 minutes. So within the 20 minutes, the particular E. coli cells, they were capable to induce the production of two daughter double-stranded DNAs here. And here the particular double-stranded DNA, here it was termed as what? Hybrid DNA. Hybrid DNA, it is an association of both N14 and as well as N15. So just look at here. N14 and as well as N15. So here why the particular hybrid DNA, it is an association of both N14 and as well as N15, because initially the particular E. coli cells, they were grown up in case of the ammonium chloride solution. Here the nitrogen which is a part of the ammonium chloride, here it is nothing but what? Heavy isotope. But here in the second strategy, when the generation time, when it was around 20 minutes, under such condition, here the specific E. coli cells, they were grown up in case of the normal nitrogenous source. So the reason why the DNA which was successfully synthesized within the E. coli cells, here it was termed as what? Hybrid DNA. Hybrid DNA, it is a mixture of both N14 and as well as N15. So here it is nothing but what? Heavy isotope containing DNA. By taking the advantage of the nucleotide sequence of N15 DNA, here the complementarity strand, it successfully synthesizes here. So just look at here. It is nothing but the complementarity DNA strand. So it is N14 containing DNA and here it is N15 containing DNA. N15. So here the hybrid DNA, it is a mixture of both N14 and as well as N15. So here why the hybrid DNA, it was successfully produced here, because initially the E. coli cells, they were grown up in case of the ammonium chloride solution. Here the nitrogen which is a part of the ammonium chloride, here it is nothing but what? Heavy isotope N15. And whereas in the second strategy, the similar equalizers, they were grown up in case of the normal nitrogen source, which is having the lighter isotope, which is nothing but N14. So the reason why within the equalizers, the DNA which was successfully produced, here it is nothing but what? Hybrid DNA. Hybrid DNA, it is a mixture of both N14 and as well as N15. Then after the successful isolation of the DNA from the equalizers, these people, they have placed the particular DNA in case of the centrifuge tube, which was enriched with cesium chloride solution. And here, the particular centrifuge tube, it was carried out for density gradient centrifugation. And as I have already mentioned you, in case of the density gradient centrifugation, by taking the advantage of density as the factor, here the DNA band pattern, it gets successfully separated. So here, because of the presence of hybrid DNA, during the density gradient centrifugation, here the DNA band pattern, it was not observed either at the top nor at the bottom, but even the particular DNA band pattern, it was observed at the intermediate position. Is it clear? Because of the presence of the hybrid DNA, here the particular DNA band pattern, it was observed at the intermediate position in case of the centrifuge tube. Here the DNA band pattern, it was not observed either at the top nor at the bottom, and even the particular DNA band pattern, it was observed at the intermediate position within the centrifuge tube. So just look at here. So let us assume it as the centrifuge tube, and within the centrifuge tube, here the hybrid DNA, it was observed at the intermediate position. Intermediate position, right? N14 DNA, and whereas it is N15 DNA. Then after, in the second generation, again they have repeated the same protocol, but even the generation time, it was doubled. In the first generation, it is the first generation, right? In the first generation, the generation time, it was around 20 minutes. So here, when the generation time, when it was around 20 minutes, under such condition, here the equalizers, they were capable to regulate the production of 2-daughter double-stranded DNAs. But whereas in the second generation, when the generation time, once when it was doubled, that means when the generation time, when it had become 40 minutes, under such condition, instead of producing 2-daughter double-stranded DNAs, here 4-daughter double-stranded DNAs, they were successfully produced. And again, here we need to recall the semi-conservative mode of replication. So during the semi-conservative mode of replication, what exactly happens here is, in case of the parental double-stranded DNA, both the template DNAs, both the strands of the DNA, they act as template. So here, the red color DNA strand, it acts as a template, and whereas the green color DNA strand, it also acts as a template. And again, here the red color DNA strand, it acts as a template, and even the green color DNA strand, it acts as a template. And in case of the semi-conservative mode of the replication, what exactly happens here is, in case of the dotted DNA strand, there is an availability of 2 single strands. One of the single strand, in case of the dotted DNA, the nucleotide sequence of one of the dotted DNA strand, it remains very similar. Likewise, the nucleotide sequence of one of the template DNA. Is it clear? The nucleotide sequence of one of the dotted DNA strand, it remains very similar. Likewise, the nucleotide sequence of one of the template DNA. So just look at here. The nucleotide sequence of the N15 containing DNA, it remains very similar. Likewise, the nucleotide sequence of one of the dotted DNA strand. So just look at here. Then in the same way, the nucleotide sequence of the N15 containing DNA, it remains very similar. Likewise, the nucleotide sequence of one of the dotted DNA strand. And again, by taking the advantage of the nucleotide sequence of this particular dotted DNA strand, the complementary strand, it successfully synthesizes here. Complementary strand, right? Complementary strand, it successfully synthesizes. So here it is N14, and whereas Here it is N15. So here in what way? In what way the N15 containing DNA it acts as a template, and even the N14 containing DNA even it also acts as a template. So here the nucleotide sequence of this particular template DNA it remains very similar with the nucleotide sequence of one particular daughter DNA strand. So here the nucleotide sequence of the N14 containing DNA it remains very similar with the nucleotide sequence of one of the daughter DNA strand. So just look at here. So here the nucleotide sequence of the N14 containing DNA it remains very similar with the nucleotide sequence of one of the daughter DNA strand. In the same way, the nucleotide sequence of N14 containing DNA it remains very similar with the nucleotide sequence of one of the daughter DNA strand. And here by taking the advantage of one of the daughter DNA strand, by taking the advantage of the nucleotide sequence of one of the daughter DNA strand, the complementary strand it successfully synthesizes here. So here it is nothing but what? The complementary strand. Complementary strand. N14, N14, and here it is N14 and N14. So after the successful isolation of the DNA from the E. coli cells, and again these people they have carried out density gradient centrifugation. And here in case of the density gradient centrifugation, here these people they have observed the N14 containing DNA which was remained at the top, but whereas the N15 containing DNA it was remained at the bottom. So just look at here. Let us we assume it as the centrifuge tube. So within the centrifuge tube, these people they have observed the N14 containing DNA which was remained at the top, but whereas the N15 containing DNA it was remained at the bottom. N14, N14, and here it is N15 and N15, right? So with this simple observation, these people came to a conclusion that here the mode of replication in case of all the living organisms it is semi-conservative mode. So once when the generation time, once when it was around 40 minutes under such condition, here 4 daughter double-stranded DNAs were produced. But once when the generation time, when it becomes 80 minutes under such condition, instead of producing 4 daughter double-stranded DNAs, here 8 daughter double-stranded DNA it successfully produced here. And likewise, the specific observation which was carried out by Matthew, Meselson and Franklin Stahl, the similar observation it was carried out by Taylor. So here Taylor, by taking the advantage of radioactive thymidine, he had performed the similar observation in case of a specific living organism termed as Vicia faba. So just let me write it here. Taylor, by taking the advantage of radioactive thymidine, he had performed the similar observation in case of Vicia faba, which is otherwise known as faba beans. And here the output which was obtained in case of this particular observation, it was remained very similar. Likewise, the output which was obtained in case of the Meselson and Stahl experiment. So here the DNA which was remained within the interior side of the chromosome, it was capable to replicate in the semi-conservative mode. So with this observation, these people came to a conclusion that here the mode of replication in case of all the living organisms, here it is semi-conservative mode. Is it clear? So here this is regarding Meselson and Stahl experiment from the molecular basis of inheritance in detail. And in the next upcoming lecture session, we will be studying regarding DNA fingerprinting. Is it clear? So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.